welcome you for our PhD webinar series organized by Microbiologist Society, India. And we meet every alternate Sunday for this PhD webinar series. But last Sunday, there was some medical issue of speaker. So I apologize all the participants who were waiting for a webinar. It was not possible to conduct. At the last moment, we have cancelled that lecture, and it will be conducted coming next Sunday. So today's our lecture number is 34, and the speaker for PhD webinar series is Dr. Anuradha Zappe ma'am. I
Namaskar.
welcome you. Also, I welcome our Chief Patron, All India President of Microbiology Society, Dr. A.M. Deshmukh sir, and all other participants. I welcome you on behalf of Microbiology Society. First of all, now I introduce today's speaker, Dr. Anuradha Zappe ma'am. She's working as assistant professor at Bharati Vidyapith University in Department of Microbiology. Also, ma'am has 22 years of teaching experience and also research experience. She has worked in National Chemical Laboratory, Pune. Also, she has published many research articles internationally and nationally. Also, ma'am has attended many conferences and workshops and training. Also, ma'am has one patent on her name. The title of invention is Intelligent Multiple Regression Analysis in Agriculture, Crop Productivity, Rainfall, Fertility, Humidity, Temperature using machine learning. So I welcome you, ma'am. Once again, I welcome you, and now I request you to start your presentation.
Thank you, madam. At the outset, I want to thank Honorable President Dr. A.M. Deshmukh sir for giving me this great opportunity to be on this platform today. And surely I want to thank Honorable Dr. M.G. Bodhankar sir, who have been motivating me.
Hello.
Yes.
Only 1 minute. I want to talk something.
Yes, sir. Please.
First of all, I want to welcome you on the platform of Microbiologist Society. Today's your topic is a really advanced topic. Many students from India will get benefit because after recording the lecture, we are going to circulate throughout the India. And later on, we observe that the views for recorded YouTube lecture increases after some time.
Right.
So we have created a very good academic environment in India, and many young teachers like Krushanjali are helping me. Enthusiastically, they are organizing this lecture series every alternate Sunday.
Yes.
I am also thankful to Dr. G.V. Mari, who is your vice principal of your institute.
Yes, sir.
He has been here to appreciate you.
Yes.
And to give importance to the Microbiology Society activity. Yes, Mari sir.
Thank you, sir.
Yes. So I request you to proceed further. Thank you.
Thank you very much, sir. Shall I share my screen?
Yes, ma'am.
Yeah. I just continue with my initial words. I wanted to thank Dr. Deshmukh sir. And the next I was expressing my thanks to Dr. M.G. Bodhankar sir, who had motivated always to present on this platform. And it is really coming true today because of Mari sir and Dr. Deshmukh sir. So thank you to all of you, sir. And I take the permission to begin with my presentation. Right. Is my screen visible, ma'am?
Yes, ma'am.
Yeah. Shall I go on presentation mode or it's okay for you?
Ma'am, on the presentation.
Pardon?
Go on presentation mode, ma'am.
Okay.
Hmm.
Just a second for me. Hmm. Yeah. Is it seen?
Yes,
ma'am.
it is
Yes.
visible.
Yeah, fine. And just I take it to presentation mode.
Yeah.
And
Yes, ma'am.
okay, actually it was an ad which is I want to run here for a moment. Kindly allow me.
Yes, ma'am. Go on.
Yeah.
A close pair of stars. चलो हो गई स्टोरी अब सोचो यही
आंखें, दो कान और पत्ती तो देहरी।
देखा
हाँ, फिर?
टूटता भी हो गया आने दो देख ले 90% ब्रेन का विकास 1 मिलियन से बोनवीटा लिटिल चैंप पिलाती हूँ इसमें है DHA
आपके जानने से पहले आपका बच्चा बन जाएगा स्मार्ट। बोनवीटा लिटिल चैम्प्स, इसमें है DHA, खास 2 से 5 साल के बच्चों के लिए।
So this is the DHA molecule, which is one of the most required polyunsaturated fatty acid in our metabolism. And my topic is related to production of these polyunsaturated fatty acids using marine yeasts. So we proceed with the slides further. Yes, so just an overview about the topic. So when it is to polyunsaturated fatty acids, of course, these are long chain hydrocarbons having carboxyl molecule at one end and methyl molecule at another end. And as we can see from the methyl end, the position of double bond expresses its degree of unsaturation, and the position indicates the type, that is at position 3. So it is omega 3. At position 6 from methyl end, so omega 6, like that we have series 9, 12, etc. However, these long chain polyunsaturated fatty acids like DHA, EPA, GLA, ARA, these are known very potential metabolites and are essential fatty acids for and are to be received from nutrition. Of course, we have the pathway that keeps converting the basic C18 molecule to these long chain fatty acids. However, there are many factors which affect this process, and desaturases and elongases enzymes are required, but for human beings and mammals, these healthy PUFAs are essential fatty acids. And as we have seen in the advertisement, it is essential for brain development in fetus, infants, cognition, IQ, and even in the aging process. If you have the deficiency of these molecules, person can suffer from the issues like Alzheimer's and Parkinson's. So right from fetal development, brain visual functioning, signal transduction, cardiovascular functioning, and prevention of cancers, etc., are the benefits of these molecules. So these are highly essential molecules. So of course, what are the sources for it? So vegetable oils, they provide us with omega-6 fatty acids, and omega-3 fatty acids. If you look at the vegetarian sources, they are really very limited. The amount that we consume also is very less. And main sources are animal sources, and major are the fish oils. Now, these are various health benefits as we have discussed earlier. So the ratio of omega-3 and omega-6, that is omega-6 is to omega-3 ratio, is a key indicator. And your index for these two fatty acid, these two group of fatty acids is very important. That dictates the status of your health. The desirable range of your omega-3 index is between 8 to 12. And for that, what you need to have is right from 250 milligram per day to 500 milligram per day, depending upon your health status, are the recommendations given by global recommendation bodies. That is 250 milligram to 500 milligram is to be consumed per day. So the problem is, do we get enough of it through our nutrition, or it is to be taken as dietary supplement? That is to be seen. Okay. Now these are the different, there is some problem in the key to run the slide. So I am switching from the PPT mode to this mode again. Kindly excuse me for that. So if you look at research on PUFA and health status, now if you go in 1960s, dietary fat was considered as a bad fat and bad for heart. However, in 80s, these fats were studied by a biochemistry point of view, and they became the characteristic indicators or biomarkers regarding the health status. Further, in 1990s, dietary composition of fatty acids was studied in details, and then good fat, bad fat, and in good fat, PUFA got the importance, and they were talked in details. In 2000, the plasma levels of PUFA got the designation as biomarkers or the omega-3 index of your health status. And of course, it is going to be dependent on various factors like personal factors, behavioral habits. Yes, we ignore it, but it is really very important and is at a challenge because of our day-to-day routine. Racial subgroups, ethnic subgroups, environmental effects, and so on. So when it comes to the sources, as I said earlier, for vegetarian people, the sources are really very limited, and the type of fatty acids that we get out of it are majorly the basic precursor molecules, alpha-linoleic acid and linoleic acid and GLA, that is omega-6. But when it's come to omega-3, these are the marine fish, and of course, on today, there are omega-3 eggs and related products on the market, which can suffice the requirement for omega-3. Now, when we go to the importance of this topic, it is to be highlighted that the ratio of omega-6 to omega-3, now in prehistoric time when human beings were hunter-gatherers, it was 1:1. And there are no records of people, human at that time were suffering of heart-related issues. Of course, it has been changing over the period of time, and as we progressed, the diet methods also changed. And in today's case, at least in India, we have the ratio that has changed from 1:1 to 14:1 in majority of population. And if you go to Western countries, it is as high as 50:1, and which is surely the important factor in inducing non-contagious chronic diseases. So if you look into the research carried out since 2010 only, if you look at the diseases and the role of PUFAs, so many % of research papers are talking about various disorders like arthritis, cardiovascular disorders and diseases, then diabetes, type 2 diabetes, hypertension, etc., the metabolic disorders in general. And here is the graph that shows the demand for these molecules as nutraceuticals or medicinal supplements. And by 2025, it is expected to be as high as 50,000 tons annual requirement, and with the growth rate of 15.7%. So this highlights the importance of this topic and the need to have alternative sources, as the natural sources are depleting. That is, the major source for omega-3, as I mentioned, are fish, and fish are being predatory on the zooplankton and microplankton from seawater. Actually, these are the producers in the natural environment, the microbes, and they are fed upon by higher organisms, and that's how it gets accumulated in these higher fish, and their metabolism, of course, supports that, but the basic source are microbes. So alternative source for every problem, as Dr. Deshmukh sir always says, are nothing but microbes. Same applies here. We have microbes as alternative sources. So why to go for microbes? There are certain benefits when we go for any metabolite from microbes. It is a clean technology, we all know. Then we can have tailor-made molecules. There are no environmental parameters or seasonal dependency and so on, which we all are aware about. And in addition to that, many of the microbes, they have antioxidant molecules produced, so they are going to be helpful in maintaining the stability of these oils. So here are certain organisms which are referred to as oleaginous, that is the organisms which can accumulate high amount of oil. Otherwise, every organism has, even in our adipose tissues, have oil droplets stored in. But when it is more than 20% of dry biomass, you can refer to it as oleaginous cell or microbe. So there are oleogenous organisms which are enlisted here, which we know are microalgae. Most of them are used for biodiesel because the type of oil they accumulate are saturated fats. There are few references from microalgae also, which produce polyunsaturated, but there are limitations against yeast when it comes to production. Bacteria, of course, they do produce. These are the different substrates being used by people for producing lipids, and these are the amounts of lipids produced. Yes, fungi and yeast are equally prominent, but amount of PUFA produced, and when it comes to fermentation processes, they are more amenable as against bacteria and microalgae. And bacterial cell size and the amount of oil accumulated may it be 30%, but see the cell of bacterial cell and see the cell of an yeast or the filament of a fungi, when it is 61% of 1 millimeter and 60% of 1 micron, there is a difference, right? So that is the beauty of yeast and fungi. Now, when it comes to the oil sources, vegetarian, as I referred to, this is from oil seeds, and here are the microbes. Among microbes, these are the yeast like Cryptococcus, Lipomyces, then Rhodosporidium and Eurovia, the workhorse in lipid chemistry. These are talked a lot, but of course, again, the limitation is the type of polyunsaturated fatty acid it accumulates. So if you look at the yeast and fungi, which are isolated from soil and fresh water, you can see they do not accumulate high-end LC PUFAs, right? So lipid accumulation is good, but LC PUFA, that is C20 onwards, and with unsaturation is ND, that is not detected. So as I said earlier, the only source is the marine microbes. Among marine microbes, yeast I had chosen. And if you look at the studies carried out on PUFA production by yeast, this is a part of a list of patents been published. And you can see all our international patents, Indian patents yahan pe dikhte hi nahi hai. And secondly, when the things are patented, they are not available to common population that easily. So as microbiologists, it's our duty to work on it and make the technology easily available to our countrymen. So the aims and objectives of my presentation were to isolate and screen promising PUFA producing marine yeasts, to identify the isolates, of course, by biochemical and molecular methods. I have done it. Then the major issue was to optimize a low cost substrate and formulate an efficient media to produce PUFA using the selected isolates, and then to optimize the production system and to prepare a biomill. That is, instead of taking the peels of these fatty acids or PUFAs as recommended by physicians, it would be always better to introduce it into the food chain. So if your milking animals are fed with it, or the fish which are reared, because most of the times fish catch from the seawater that is so polluted is coming to us, and it is very rare and banned at certain sites. So most of the fish that reaches to market are the pond reared fish, and they are really lacking in NC-PUFA. So their feeds also should incorporate these molecules. So the idea was to prepare a biomill which can be converted as per the requirements of the animal husbandry that you are looking into. For higher animals, milching animals, it will be different. For fish, the particle characteristics, etc., are different. But basically, to produce it in biomill form was aimed so that we can bring it in the food chain, and through animal sources, through their products, it will be available. So these are the highlights. And of course, it was very mandatory to check the toxicity of the product biomeal that was produced. So in overview, 3 lines: isolate potential oleaginous PUFA producing yeast, design the low-cost media using the waste bioproduct, like dairy waste. I had used whey. Then I had tried to use all other substrates also were tried, but basically I tried to valorize whey, dairy waste, that is the whey, and prepare the biomeal that can be used as feed supplement. Is it going okay? Hello? Hello?
Yes, ma'am, it is fine.
Yeah. I thought it is fast, so I just wanted to know. So when it comes to yeast are as promising LC PUFA source, 20 to 80% of of their dry biomass they can accumulate. So they are really very promising, and the cell size is also good. So Of course, lipid content and the lipid profiles, they differ between species. And of course, what substrate you provide accordingly, the lipid profile also will change. Another thing is yeast can utilize variety of carbohydrates, renewable sources, industry byproducts also can be used by these. And they are little more easily amenable against bacteria or other specific organisms when it comes to osmotic stress condition. That is, they can tolerate better than bacteria. And their survival rate also is better in dry conditions. So growth rate also, if you compare against filamentous fungi, of course, filamentous fungi do accumulate equally good amount of oils, but extraction from filamentous fungi is a very difficult process as against that from yeast. And the growth rate, if you can compare, yeast are far better than filamentous fungi. So yeast were chosen. So this is the Indian Konkan coast. From the untouched beaches, I had taken the samples and had tried to isolate the yeast from it. So this is the general overview of the methodology in the primary screening, secondary, tertiary screening, then preparation of biomeal and toxicological study. In primary screening, I had done microscopy, gravimetry, and biochemical characterization. A paper was published. Secondary screening was molecular identification and confirmation of type of oil produced by GC and FTIR. And of course, media optimization studies, which is going to be a continuous process till you hit the best possible media. Then tertiary screening, I had tried to check whether the yeast are comfortably producing the desired PUFA in submerged fermentation or solid state fermentation, and the comparison was done. And in the next step, the solid state fermentation methodology was optimized using response surface methodology. And the biomeal produced was scaled up at laboratory scale, and that was fed to Vista rats, and toxicological studies were done at National Institute of Toxicology. So going to the overview of results. Now, as we all know that fatty acids present in this, fat accumulation or lipid accumulation in any living cell or dead cell can be easily detected by basic microscopy techniques. All of we do it in FY, the Sudan black bee staining method and wet mount. So this particular slide is showing in this corner is the wet mount of a yeast cell, which has accumulated. These white globules are nothing but the oil droplets. And over the period of time, these small droplets, they coalesce, and a very big size droplet you will see, which fills the entire volume of the cell. So this is the reference picture which I had taken from this particular paper, and these are my yeasts. I had initially I had isolated about 56 yeast, out of which 11 were shot down owing to their oil accumulation capacity, and then I had concentrated on 3 of them. And I'll be talking about their identification, but these are the pictures of the yeast which I have played with. So here, this is the wet mount, which is showing these white droplets. So these pockets are nothing but oil droplets. Now, Sudan black bee staining, we know that the oil globules get stained by with bluish black color, right? So if I try to stain my marine yeasts with the standard Sudan black bee staining method, this is what I am seeing. So if you see this picture, it is very clearly seen that the inside the cell, nothing is seen, irrespective that wet mount is showing me the pockets. The problem was penetration of Sudan black bee inside marine yeast. The cell membrane and the peripheral membranes of the yeast cell were not conducive for penetration of Sudan black bee. So I attempted to modify the age-old Sudan black bee staining method, and certain pre-treatments were given to these yeast cells that would allow penetration of Sudan black bee. And this is the publication out of which now you can clearly see the difference. This is with the standard Sudan black bee staining method, and this is stained with the modified method. So this was the primary screening to locate whether the cells are accumulating oil or not. So at this stage, I could get another publication. This is the publication. It is in the Scopus Index journal, entitled as International Journal of Current Microbial Applied Science. It was in Scopus at that time. I don't know the present status. Now, these are the different yeast, the 3 standards which I had taken further. These are the laboratory titles given to them. We will come to their identification as well. So you can see it here that at every stage of growth, that is 24 hours, 48 hours, 96 hours, 120 hours, 196 hours, till the cell was accumulating oil, I had screened those and I had stained those cells at every growth stage and accumulation of lipid. As you can see, the size of the blue color pocket here. See the size here, see the size here, and see the size here. It goes on increasing. See, this particular cell is almost full of oil. So with being the secondary metabolite, after the growth is completed, masses of this oil get accumulated. So this was the primary screening part done. Now, gravimetry was essential. That is how much actually is quantity of the oil produced. So Soxhlet extraction was done in hexane, and the amount gram per dry biomass of the cell was analyzed. For all 11 yeast, I had done this. Here, characterization of the isolates is to be done. So initially, for characterization, it is really important that we need to know dry biomass, absorbance and colorimetry, and colony forming units. So for all these heats, I had collected the data which is relevant. Interplay of these three factors was characterized. So these are the graphs which show the absorbance, dry biomass, and weight and CFU for each of them. Second factor was the pattern of glucose utilization, biomass amount produced, and the lipid accumulated at different growth phases, that was also characterized. Now, this particular part, that is absorbance measurement, CFU and DVM, is very essential because whenever we go for biochemical characterization, the amount of cells going in per unit tube, that is the test tube for characterization of, say, glucose, rhamnose utilization, etc., it should be uniform. Otherwise, we are going to get false positive results. So the quantified amount, every test tube had quantified amount of inoculum. Number of cells were measured and they were inoculated. So these are the different sugar utilization tests they were done. And of course, marine yeast need to be observed for their biochemical test for 21 days. It is not the 48 hours or 72 hours experiment. Every day till 21 days it was observed, because the patterns of utilization also change with time. So considering the references shown earlier, I had observed it for 21 days. And then after these different sugars, that is try to complex polysaccharides, other things like succinate, citric acid, lactic acid, and glycerol also were tested. Now, as you are seeing here, spore staining also is one of the criteria. Most of the marine yeast are asporogenous. And here it is hemolysis assay as a preliminary screening whether the isolates are having any toxicity to the cells. So the hemolysis assay was done. So by here, biochemical characterization had told me that is oxidative mode of sugar utilization, which is the very preliminary and important characteristics of marine yeast, because many a times human activities on seashore introduces various organisms into seawater. They dwell there, but they are not the seawater original or indigenous flora. So it was a challenge to find, and this is one of the very important characteristics, that is sugar utilization by oxidative mode. They are unable to utilize rhamnose melizitose. They are unable to utilize nitrate form of nitrogen. They are mostly asporogenous, and amyloid compounds like amylase acid, we do, it is one of the examples. So no amyloid compound secretion by these organisms was observed. The next step was to characterize them. So these are the scanning electron microscopy studies I had done. Of course, it was, I'm showing here only for the three, which I have taken further for the biomill production. So this is Candida orthosilopsis. This is Candida tropicalis, and it is Rhodotorula mucilaginensis. However, these names, they appear to be that of pathogenic fungi, but marine dwelling are not pathogenic. Even when we say about Candida, the very first pops up in the mind, Candida are the nuisance organisms, but only 30% of them are pathogens, rest are non-pathogenic. And in marine environment, of course, I could fortunately isolate these non-pathogenic organisms. Now, coming to their molecular identification, yes, 18S rRNA gene sequencing was done at NCCS. I am thankful to Dr. Yogesh Shauche sir, who had made this facility available to me and had given an opportunity to work over there. I have published those 11 databases with the elegant team of, legendary team of NCCS. And they are on the gene bank NCBI site. So these are the three which I have taken further, that is of Candida tropicalis, orthosilopsis, and Rhodotorula species, which I have worked with. Now this ends the initial screening process. Now it was required to characterize the oil produced. So initial studies were carried out on TLC. And here, as you can see, this is the solvent system that was used and detection system used. So this is the origin point. And from here you have monounsaturated diacyl glycerols and triacyl glycerols, and free fatty acids also can be located. The diagram is a little difficult to visualize, but other diagrams further are self-explanatory. But here it is from origin to TAG, DAG, monoacyl glycerols and free fatty acids depending on their length. And of course, some unknown spots are seen, which may be other metabolites which are this solvent soluble and are related to lipid in nature. So they are also seen here. So the initial screening was done by TLC. Then I had taken it for Nile red staining, which is specific for neutral lipids. So neutral lipids are mainly diacyl glycerols or esters formed, TAG, etc. So these are the three yeast which I had studied. This is the elongated cell filamentous one, the tropicalis, Candida tropicalis, which is showing the dimorphism, and depending upon the growth conditions we provide. So these are the small golden fluorescence of Nile red is observed when you give the excitation of about 450 to 490 nanometers. Excitation is seen at about 528 nanometers and above. So golden fluorescence indicates presence of neutral lipids. So you can see these shiny golden cells which were observed under this axisoscope, and the excitation filter, as I said, was 450, 490 nanometers. So this was a microscopic identification, and this facility I had explored from Department of Biotechnology, University of Pune. I'm thankful to Dr. Mrs. Jinzade who had made it available to me, and the paper published also, I have acknowledged them. Here comes the secondary screening based on FTIR studies. So these are the FTIR peaks. So initially the biomass was obtained, it was dried, oil was extracted by Soxhlet process, and then FTIR analysis was done. It was done in our college itself. I'm thankful to Dr. Dhapte sir who had made this facility available to me. And the scanning was done. Here, this is the standard lipomyces strain as control I had used, and the screens of, the pics which are you are seeing in other two photographs are the studied isolates. Now here in the next slide, it is more simplified fashion. So I'll share the next slide, which is more explanatory. This is the growth phase of the organism at 24 hours, 48 hours, accumulation of oil seen in the staining method, and it is integrated with the FTIR peaks. So if you see the peak at around 2800 to 2900, it is associated with methyl group, which is present. The one near 1700 range, that is 1670 to 1820, indicates C double bond O esters, carboxyl esters, which is fatty acid esters, the stretch for that. And here it is for the CH2 and CH3 groups. Now, very important for me was to see these two, that is methyl end and the ester end. So as you see, 24 hours, 48 hours, up to 196 hours, the FTIR scans were taken. And as you go on increasing the age of bioma for harvesting the biomass, the area under the peak goes on increasing. That is, the amount of lipid goes on increasing. So at around 1745, you see the specific stage for ester functional groups in this particular case. The range is between 670 to 1820. In my case, it is 1745. Shall I continue? Next step was I am seeing some chat. So is it relevant? Do I need to look into, or I shall continue? You kindly ping me if it is related to the topic, huh? So next was characterization by gas liquid chromatography. So just by FTIR, I could confirm the presence of functional groups, but I need to know which fatty acids are present in my cells. So of course, MS is required, but that time only GC was available. MS facility I couldn't get. So initial qualitative characterization was done with gas liquid chromatography. So this chromatogram is the standard chromatogram I'm showing you, where you have a standard of mix of standard fatty acids, and that is run, and then you have to run the sample oil, which is derivatized to produce fatty acid methyl esters. So FEMS preparation is an additional step required here after you extract the oils. So fatty acid methyl esters were prepared and they were introduced into GC, and chromatogram was obtained. So it was to know what is the amount of saturated, monounsaturated, and polyunsaturated fatty acids, and what is the ratio of omega-3 to omega-6 in the oil which I have obtained from my marine isolates. The ratio is pretty good, right? 3:1, 4:1, 0.77:0.1. So they are really promising in terms of the oil they are producing, the quality of oil they are producing. This represents the area under the curve in the chromatogram and its equivalence in terms of grams per liter, and the dry biomass of the cell is expressed in the chart. Now coming to the point, by now whatever we had done was microscopy to isolate the oil accumulating cells, then analytical methods for TLC, FTIR, GC were used to check the presence of PUFA in the oil and its content. Then marine origin of the yeast was confirmed, and the isolates were confirmed by rDNA sequencing, and they were registered at gene bank. Now next was to see how do they perform in vitro, because when it is to fermentation or large scale production, optimization is a great challenge, and that was the next part of it. So initial, as I told you, whey was used as the basal media component. And to that, I had supplemented with various carbon sources, because carbon nitrogen ratio is very crucial when it comes to accumulation of lipid by de novo synthesis method. Ex novo, that is what you supply in the form of oils, is taken up by the cell, and then they accumulate their own oils. And de novo synthesis is utilizing the carbon available. The metabolic pathways are diverting to accumulation of lipids. So that is more feasible because low cost substrate is to be provided. So of course, starting from glucose, I had used flax, glycerol, etc., and a series of various carbon sources, including tomato waste, then orange fruit peel. These are also the good known sources. So the next slide will talk about it, but it is just to tell you what was done in optimization of media. Various carbon sources in various ratios and combinations were analyzed to see the response of these three isolates and the amount of oil produced and the ratio of saturated fatty acids to monounsaturated and to polyunsaturated fatty acids. So as you are seeing in the graph, there are 3 bars per analysis. So each graph represents each type of fatty acid present. So every time the entire process till GC was repeated, from extraction, growing, extraction, then derivatization and GC. Hundreds of GC I had performed to come to these decisions. And here are the patterns, that is the accumulation of desired fatty acids. As I said, DHA and GLA was observed, gamma linoleic acid. Of course, though it is omega-6 fatty acid, is a very, very important molecule because it is the precursor. Omega-6 also are the precursor for signaling molecules. So like prostaglandins and leukotrienes and thromboxanes, etc., etc. So omega-6 message GLA was observed in my cells. So this is the pattern of accumulation of these two metabolites. So see, it is number of days on x-axis. So 0 days, 3 days, 10 days, 15 days are on the scale, and area of oil. So GC mein, what is the area under the peak of total oil and for respective oils that was taken, and the graph is plotted. So this is DHA accumulation, which goes on about by 5 or 6 days, we get the peak. All right. And when it is glycerol is used as substrate, when flax was used, I could get high amounts by second day only, but the quantity accumulated was pretty low. So ex novo synthesis was not that beneficial. De novo synthesis I had to concentrate on. So these are the representative graphs. So this, like this, for all other carbon sources, it was studied. So it is similar to that, and it is a summary of all the parameters, all the carbon sources that I had used. As I said earlier, so yeast extract, glucose, then tomato waste, orange peel, groundnut oil, safflower oil, flaxseed oil, and the cakes also were used. After extraction of oil from the seeds, the cake that remains also was tried as a substrate, because the whole idea was to focus it as low cost technology. So for every media combination, all these 3 yeasts and the interplay of these parameters was to be studied. So here in every media combination, you are seeing 3 bars. Of course, again, they are MUFA, saturated, and PUFA fats and total oil that is talked about. So it was screened. So again, it was not only just on the basis of a GC graph. The biostatistic analysis was done by Duncan test, And these are the plots obtained after statistic analysis. And as you see, depending, these are the isolate names. So for every isolate, the priority for the carbon source is little differing, but yes, commonly glycerol and whey are giving a good amount of response as equivalent to 30% glucose. So glucose is of course pretty refined and costlier molecule when it comes to large scale. So I had tried to opt for the other sources. And of course, whey is a crucial player in all of these. Now, these are the optimizations of the summary of optimization done. That is what time hours required, at what temperature it performs better. And depending on temperature of incubation, type of fatty acid accumulator also, and the quantity also varied. All these studies were done. And then all these studies were done in submerged fermentation, right? But we know how solid state fermentation outweighs submerged fermentation. If it is possible to do, of course, we shall go for it, considering the benefits associated with solid state. So I had tried to check whether my yeast are comfortably growing in solid state fermentation. So these are the inoculum prepared. I had used corn as a substrate, carrier substrate, because corn is one of the important feed component, right? And of course, if at all the residual oil in the corn again is going to be beneficial for the animal that is consuming it. So feed supplement, for feed supplement, corn I thought was the best carrier. So that was used, and biomeal was produced by making the powder of dried corn and inoculating it with the inoculum of these 3 yeasts. And the characterization again in the similar way it was done. Like for every parameter which was checked for submerged fermentation, I had checked it for solid state fermentation. And here are the comparative graphs for all the 3 detailed studies. That is in submerged against all these media combinations in high count, and the PUFA content went on increasing, accumulation inside the cells increased with incubation time. So I decided to concentrate on it. This is the paper published on the basis of that. Now here, it is very required to optimize every parameter when you are claiming some media as the optimum media. And generally we go by OFAT method, but when it comes to large scale combinations, of course, surface response methodologies are important. So I had developed a CCD matrix. Factorial design was developed. That design matrix software was used for it. Multiple regression analysis of the data generated was done. And the equation generated for GLA production by Candida tropicalis, I'm sharing here with you. I'm not going to go into the details of it. A paper is shortly coming, so I cannot disclose everything on it. So these are the contour plots, etc., which are often. But in short, based on this response surface methodology, I could get a combination where 0.1 gram % yeast extract, and then pH 5 and glycerol of 5% with whey base and corn as a carrier could produce very high amounts of LC-PUFAs. So that was used further. And the biomeal was produced at higher scales. That is, initially it was for 10 grams, then for 100 grams, and it is in kgs. And this biomeal was characterized in terms of cell count, protein content of yield by Geldan method, total oil, then a ratio of different types of oil, that is MUFA, PUFA, and saturated fats, and by GC, the specific GLA and DHA content present in the oil. So once that biomeal was ready, it was very mandatory to go for toxicological studies. So the toxicological studies were done at National Institute of Toxicology, Dhairi. So Vistar rats, 6 groups were studied. Of course, it was done according to OECD guidelines 407. The rats were fed with the biomeal every day for 28 days. It was 28 days subacute repeated dose studies. And at the end of that, animals were sacrificed, and serum biochemistry, hematology, and histopathological studies were done. And fortunately, there were no abnormalities, no variations no signs of toxicity or trouble were seen. So in conclusion to say, I had isolated 56 out of which 16 yeast which were promising potential PUFA producers. These marine yeast were characterized, identified by gene sequencing and are deposited at NCCS and the sequences at GeneBank in CBI. Whey-based corn meal was used as a substrate for PUFA production by SSF and whey-based medium for submerged fermentation. Then submerged fermentation was replaced by solid state, considering the benefits associated, and the system was optimized for production of LC PUFA having the biomeal having LC PUFA or the biomeal enriched with PUFA. So nutritionally enhanced biomeal with GLA, DHA and other LC PUFAs, good amount of proteins, and in case of rhodotorula, the pigment also. So that was prepared. The study, toxicological study of the formed biomeal was done in Vista rats, and it was found to be safe as feed supplement. So the future work, which is a part of it, is in progress, but yet not to the speed which I want. That is to check it in flocks and fish, to check the shelf life of this biomeal, to develop a ruler technology for easy production of this biomeal, etc. And yes, in your business, performance is crucial in ours too. So it's a cartoon which narrates a lot. And with this, it is the summary of my presentation. Here are the certain references. And these are the outcomes. Best Poster Awards. I had got 4 of Best Poster Awards for these presentations, a few Scopus Index publications, radio talks, and UGC minor research project. Also, I had completed on this. I'm really thankful to the authorities of Bharati Vidyapit, Dr. Kadam sir, the former BCUD director Bulak sir, Budhankar sir, my guide, Dr. Mrs. Sapre, and Dr. Abhay Sulkar, who had given me total free hand for independent working. So that is very required when a student wants to explore the ideas. Conscious, free hand is to be given, and I'm really fortunate to have these 2 guides. And of course, my sincere thanks are due towards Dr. Shauche from NCCS and the director of MCC, Microbial Culture Collection. He raised that department institute further. Dr. Apte from National Institute of Toxicology, and of course, Dr. Kulkarni from Kulkarni Laboratories, where I used to go for extraction of oil from my yeast cells. So every cultivation, I had to go for extraction till my minor project was sanctioned. Stoxel extraction was done at his lab, and protein analysis also was done as his lab. So I'm thankful to him. And this is all from my side. I think in time I'm able to conclude it. Yeah, there are 3 questions, like why selective screening of oleaginous yeast was not carried out. Selective is to towards a type of specific PUFA. Of course, unless you go on for checking the amount it produces, it may produce a very good amount of desired PUFA, that is high DHA producing. But if it is not propagating at a faster rate, the growth rate, the amount of oil it accumulates, if it is less, then it is of no use. So initially it was mandatory to check the oleagenicity of the cells and then to go for the selective screening for desired PUFA. I see the second question, that is about patents. The patents, yeah, they are pertaining to, most of them are pertaining to engineered strains. A very few they are there who talk about natural isolates, like you all know, that is Aerovia lipolytica or Lipomyces starkii. These are the known organisms which have been explored by many of the companies, the great companies in biotechnological area. So yes, some of them are engineered and some are native species also. When it comes to example like Aerovia lipolytica, it is good producer of lipids, but it produces saturated lipids. So DHA or other LC PUFAs are not produced. So bioengineered Aerovia is there, and so the patent is on that. Rhodotorilla is known for production of carotenoid. How biosynthetic pathway for PUFA differs? Of course, again, carotenoid pigments, if you look at the basic molecule from where the biosynthetic pathway begins, are acetyl-CoA, right? So carotenoid is that 5-carbon basic monomer is there, which goes on into an addition head to tail or head to head, and then you get on elongation of the chain that gets converted to carotene, and then further to vitamin A in higher organisms, right? But the basic molecule is acetyl-CoA or oil accumulation as well as carotenoid. And of course, in Rhodotorilla, the carotenoid, though it is a secondary metabolite, it accumulates by end of 48 hours. It's natural shield, you can say, that gives the stability to the oils accumulated, antioxidative stress and other metabolic reactions. So at the base, you have same common molecule. See, there are many species of Candida and Rhodotorilla being studied and mostly been talked a lot about as pathogenic. But when it is to, of course, not only for human beings, but marine organisms also, you have pathogenic Rhodotorilla, pathogenic Candida. But as I said earlier, among Candida, only 30%, not even 40% known Candida species are pathogenic. Rest all are non-nuisance causing. Same applies to Rhodotorilla. And that's why initial screening was done to check the simple was initially was hemolytic acid. And for the toxicological stage, the study is a higher version of studying about the pathogenicity of the organism. So there was a challenge to select the non-pathogenic ones. And initial screening went on for more than 1.5 years. I have in total work for 7 years to complete my studies. Isolation of those yeast initially took hard time, as I told, screening for their biochemical characteristics, simple sugar utilization test, for each test for 21 days to be observed. So screening is really challenging to confirm because most of the pathogenic organisms, they are fermentative in mode. Acid and gas production is there when they get glucose and all. So certain these criteria which are shared on the screen, they are the highlights that talk about marine origin, and further on the toxicological studies done, they talk about non-pathogenicity. Yes, please. I think I have answered the question seen in the chat box. Yeah, as I said, yeah, I see why why corn is used as substrate. Yes, I mentioned during my presentation that is corn is a known feed supplement, and my aim was to develop a feed supplement. So it would be a good if it is instead of fortifying your feed with the molecule I'm preparing, it would be better if the feed itself is used as a carrier. So corn was used, and corn also is a good producer of oil. So if at all de novo synthesis is also accompanied by ex novo synthesis by these organisms in depending upon environmental parameters, it would support corn would support and provide oil source. Generally, what is done is fortifications are done in the feed, so that dosage and the quality, the pricing, all these things they increase. So if it is developed as आपण जसे सांडगे बनवतो ना घरी त्या पद्धतीने जर काही बायोमिल बनवता आलं तर आणखीनच सोपं होणार आहे. त्यासाठी जे इझिली अवेलेबल आहे, जे कॉमनली फीड सप्लिमेंट म्हणून वापरलं जातं, या आणि एक्स नोवो सिंथेसिस जिथे पॉसिबल आहे असा कॉर्न होता म्हणून आय हॅड ऑप्टेड फॉर दॅट. Hello, Krishanjali madam. Any more questions?
Hello.
Yeah.
Ma'am, have you completed the questions from chat box?
Yes.
Okay.
And as the time was up to 11 o'clock, so I tried to finish my presentation 1 minute to 11.
Okay, thank you. Participants, anyone have questions?
I'll
They
try
may
to
ask
answer my
directly.
best. Yeah.
They will. You may
You
ask
can-
directly.
Directly, or you can text me later on. That also is welcome anytime.
Oh, okay,
Here,
ma'am, you share your email ID and
in the chat box. Right.
yeah.
Yeah, I'll write.
Hello.
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 Yes, sir.
One more question.
Please, sir. I'll try to answer if possible.
Good morning.
Good morning, sir.
Actually, my work is on lipase enzyme. Lipase enzyme.
Yes.
Lipase have many prominent applications. I like biodiesel, bioremediations, bioligation. I worked on biodiesel.
Okay.
I used soybean oil, olive oil. All oil are contain more than carbon, 16 to 18 carbon. Carbon number is 16 and 18 carbon.
Yeah. Yes. True.
Like
Palmitic oil and stearic oil.
olive.
Yes.
Okay.
Yes.
So can you suggest any regarding yeast? What is the role of yeast? Any yeast, any other, Saccharomyces cerevisiae produce saturated and monounsaturated fatty acids of 16 and 18 carbon, and no PUFA, no polyunsaturated fatty acids with more than 2 double bonds. To
Right.
any
Yeah, I had shared in my slide only the. I'll try to reshare it. Let's see. Those from fresh water and from soil, the yeast and fungi, they have been characterized for their PUFA content, and majority of them, they do not have unsaturated fatty acids as compared to the saturated fatty acids they accumulate. This slide, sir. At least we have from filamentous fungi as well.
Okay.
And the slide before this, it talks about yeasts also. Yeah, this one. So many are there. It is highest is 18.3.
Okay.
So you have many species of strains of, or even species, I would say, Cryptococcus and Lipomyces starkei also what we talk about. So it is producing up to C18, and rarely they have up to C24. Certain species of that Lipomyces, and among species the specific strains. So hardly 6% they accumulate higher fatty acids, as against that of 18 and 66% of total lipids.
Okay.
Even Aerovia accumulates a lot of saturated fatty acids only. The engineered Aerovia is looked at as a PUFA source, but Aerovia otherwise is known as source for this biodiesel.
Okay. Any other yeast strain which is involved in esterified, trans-esterified?
So trans-esterified are not really good fatty acids, no. So generally when we have LC PUFAs, and if they are heated or are exposed to room temperature, they get oxidized, and at high temperature exposures they get converted to trans fats. So trans fats are not really that good when it comes to LC PUFA and health.
Okay. Thank you, madam.
Thank you, sir. Thank you.
Sir.
And nice to hear that you are from the same area of work, same domain.
Okay. Yes, madam. Okay.
Of course, and my yeast I had characterized for their enzymes also lipases. Yes,
Okay.
they are
Lipase,
good producers of lipases, because I was trying to see whether these organisms are able to go for ex novo synthesis, that is taking the oil from substrate. So for which they need lipases.
Actually,
And
my
then it will be,
okay. My work is on Bacillus.
okay.
Bacillus thuringiensis.
Okay, great.
Okay.
So lipases studies also I had done, but I have not presented every work because time limit was there.
Okay.
So yes,
Thank you, ma'am.
some of my yeast are really very good producers of lipases. म्हणजे अख्खी प्लेट त्याच्या क्लिअर झोननी भरून जात होती.
Okay.
High amounts of lipases they did produce.
Okay, madam. Thank you, madam.
Thank you. Thank you.
Is there any questions from participants? I think so, ma'am. There is no more questions.
I hope it was received at your end. I was in a hurry to complete, so probably I had gone a little faster.
Okay, it's okay.
It was okay. Okay, thanks.
Okay. If participants will be having any doubt, they may ask.
Right,
Ma'am
right.
has shared email ID. So now it's time to thank you, everyone. First of all, I thank you, today's speaker, our Dr. Anuradha Dape ma'am. She has given very nice insight of her topic, and also all other participants who are been with us from last many lectures and making this webinar successful. So thank you everyone from Microbiology Society. I thank you everyone.
Thank you. Thank you, ma'am.
Okay.
Thank you, sir. Thank you, Mari, sir.
Now, ma'am, you may end.
Yes, yes. I'm leaving the meeting. Thank you, madam. Thank you for
Thank you.
your consistent presence and updating me on.
Okay,
And also thank you to Pawar madam, Ashwini Pawar madam.